Interesting. Yeah, I'm not sure. That's a good question.
Well, I'll ask you for next time because
Yeah.
I've always spent like, I was sitting in the car looking for it.
Sure thing.
I didn't realize it was this cold until I came
I
out.
know, it was very cool.
I'm not sure if I need this in here, but just in case.
All right, and I know Becky will be joining us.
I was reading them and I was like, why did I write so much on this lecture?
You got a lot to say. It's important.
I'm not sure. It felt like a bit repetitive when I was
Mm,
reading it today,
gotcha.
but after a while it's done. I'm not gonna
Yeah.
start changing the script right now.
Yeah, we'll get through it though. You'll be fine. All right. Enough. All right, now I'm just gonna wait for Becky to get here. So.
You know, this is a weird time to come because I live on the other side of town. So if I come now, I come early, and then if I leave 10 minutes then I'm later, I'm gonna be here 10 minutes late. That time of the day, the
Yep.
traffic gets really busy.
No, I understand completely.
Do you live around here?
I live about 10 to 15 minutes away. So right at the corner, like right at Westside Park, that area.
Okay. We have
So
a few schools around that.
yeah, yeah. So I just take 8th Avenue all the way down to Waldo.
That's good.
Yeah. So I make it, but I also aim to get here early. So I get here between like 6:45 and 7:15.
Oh, that's good. Good time to
Yeah.
come.
Yeah, it makes it so much easier and not have to deal with traffic. So
And then you leave earlier too, so then you don't catch the traffic
Exactly.
going home.
Yeah.
I know what you mean. And I like the early, but yeah, this is hard for me. I live in El Plantation,
oh
So-
yeah,
southwest.
yep, no, absolutely. Do you just take
I took Williston, yeah.
okay, yeah, Williston, and yeah, it's but there's definitely some annoying lights in a couple schools or like one school.
Yeah, but it's better than coming across
Oh.
town.
Absolutely.
Um,
And like if I ever need to go to Sam's Club after work, I just take Williston and then cut north.
Yeah.
It's so much easier.
so they closed the old Sam's, right?
Yeah,
Once they opened the new one.
Yeah,
they
yeah.
closed The other one.
yeah, a few several years back.
I was happy when they opened the new one because
Oh,
it's closer to
of
me.
course.
Before it was gonna be a trip.
Yeah, it's definitely a very crowded area in Butler Plaza,
Oh,
but
right now.
it is for me. It's really nice to have Sam's Club right next to Walmart. That's a nice pair. And then you
They're
have a
kind of the same, right? I didn't
Yeah.
know that they own.
one just sells it in bigger quantities.
Walmart is always so busy. It's incredible.
Yeah,
It's always, especially now around the holidays.
oh yeah, for sure.
But some of the stuff is so much cheaper. It's incredible.
Yeah, which sometimes I'm a little worried, but sometimes I'm like, that's fine.
But most of the products are this. If
Yeah.
you just compare the same brand, they are even cheaper.
Right,
The
right.
problem is, I don't think they fill their stock
hmm
quick enough. So sometimes you go there and
Right, they don't have
they
it.
don't have
Yeah. Yep. No, absolutely. And then, yeah, but I mean, thankfully there's every store you can want in Butler Plaza. 'Cause there's two Publixes, and then, where's that one? Then Walmart, and then Aldi, and a
Yeah,
lot.
I'll leave there.
I just appreciate that they put the that back road in between Walmart and
Sam.
20th Avenue up by
Oh.
that gas station. So now that's connected. That alleviates some of the traffic.
Yeah, but I hate that turn into the 20.
Oh, for sure. It is bad.
they have to make that two lane for, connect
Yeah.
Butler Plaza and Old Small. Because even when I come back, but that's my road to come home
Mhm.
every day. And that part is always packed up.
Yeah, it's such
And
a
I know
bad
there is
intersection.
like a beach, what is it there? Like a stream or something going there. So I know it's a natural area, but they will have to find a way to make it all night because
Yeah.
that part is always packed up.
Yep.
Every single day.
It's a very high traffic area.
It's a lot of student apartments there, so.
All right, we can start getting ready. I know Becky is planning on joining us, so she will be here momentarily. But until she's here, we can at least get everything else ready.
Okay.
All right. I will make some adjustments, obviously.
Yeah, I think I'm gonna keep my jacket on for now. Maybe for next one I take it out. I'm still. I don't know why I got cold. Hair. My hair is coming up. I'm taller today. I have bigger platforms.
That is okay. The mic is completely adjustable, so then I'll scoot this over. Ta-da.
Still showing there.
That is all right. I will make it disappear.
You can play your magic with
I
that.
can.
Oh, there's so many. What is happening?
I was missing my stylist.
Oh, I'm so sorry. Hello. Happy whatever this is.
How are
Very
you?
nice, very nice. More water for you.
Oh, thank you. I always
Yeah.
bring my thing just in case.
You know 8:00 is early for you, isn't it? Have you done 8:00 before?
No, it's the
Oh.
first time.
Okay, very good. Are you
And
happy
I
about
don't
this?
like it. I mean, I'm an early person. It's just I have to drive across town.
Oh, me too. I live on the other side of
Yeah.
Tower.
Yeah,
So I
maybe
drive- I
Hill
drive-
Plantation. Yeah.
Yeah, I drive into like the fireball
Williston.
now on Newberry when I leave in the morning. The sun coming down the road.
Yeah.
Yeah, no, it's
But-
anyway.
No, I mean, I'm an early person. I wake up early. The problem is the traffic in the
Right.
morning. I hate it.
If I leave my house by 6:45, it's not bad. Once the school buses are out and everything, it gets a little trickier.
Yeah.
So I just
People
want
get tricky.
people outdoors. Anyway, we're both in our basic black today.
Yeah,
so
of
I'm
course.
feeling
I was
it.
cold this morning.
Yeah.
I actually didn't think it was gonna be cold, so I had to come back and grab something
good,
to
very
put
good,
on
very
top.
And you look good. The hair. And
My hair?
And I'm just going to peek under here to see that your collar's out evenly on both sides in case we see it. And it is. And now a little bit more right here. Is it 8:00?
It is.
Oh my goodness.
I'll do it. All right, I'm gonna make sure no one disturbs us. All right. So, I should load the script. That would probably be helpful. Open.
Yeah, you don't want me making up stuff.
You see it. You see it.
Come on.
You don't want me making it up. Yes, we kind of do. That would be awesome. You know all the words. That's
Yeah.
for sure.
I was telling him that I don't know why I wrote so much on these slides. I was tired of just reading them. Like, what was I thinking? It's weird. It goes the
you see
other
it.
way.
You see it. You see it. Very nice. Very nice. Very nice. Very nice. Out of there.
Oh, make
Are
sure I
you
can
back
read.
next week, or this is it for you?
This is it until
After
December. Yeah,
Okay.
5th.
Good for you.
How's that font size? Would you like it larger, smaller?
That's good. I can read it.
All right. Could you read the first two sentences for me, or an audio check?
It says, "Hi everybody. This module we will go over some of the immunomodulatory properties."
What is that word?
Immunomodulatory.
I don't. Wow, that's an impressive
I'm
word.
telling you, I don't even, it's impressive at any time of the day, but at 8:05 in the morning, goodness gracious.
Immuno. So you say immunomodulatory.
Immunomodulatory. 8 syllables?
8 syllables.
Immunomodulatory. Yeah, in microbiology, we like long words.
You sure do.
I totally. All right, so I'm gonna back this up one slide. Start the recording. We'll leave it there for a few seconds so the editor knows what they're editing. You can go ahead and click to the next slide. Make sure that clicker's working. All right.
Okay.
And we'll do a nice little color check. Microbiology.
Immunomodulatory.
Immunomodulatory. Right, Roman clear.
I'm sorry, Isaiah. How's your immune system working
My
today?
immunomodulatory properties of probiotics.
How is your immune system working these days?
Oh, goodness.
All right. Feeling ready to give it a rumble?
Yeah, I'm ready
You're gonna
to.
do awesome.
I'm ready to be done.
No worries. Well, you can't be done until I'm ready.
Okay.
So we're gonna go.
Oh.
All right. And with that nice big smile, you can start after 5, 4, 3.
Hi, everybody. In this module, we will go over some of the immunomodulatory properties of probiotics and the bacterial components that mediate those responses. It is expected that at the end of this module, you will be able to list the main bacterial cell components involving host immune responses, list the microbial components sensed by host pattern recognition receptors, or PRRs, and to describe the effect of components of probiotics on immune cells. The beneficial effects of probiotics are believed to be derived from its ability to enhance the intestinal barrier function, inhibit pathogens, increase the abundancy of microorganisms, and by modulating local and systemic immune responses. In this module, we will focus on specific bacterial components that have been determined to act as effector molecules on these mechanisms. This complex figure is a comparative overview of the bacterial surface effector molecules that have been described in probiotic Escherichia, Lactobacillus, and Bifidobacterium. While lipopolysaccharide, or LPS, are the major component of the outer membrane of gram-negative bacteria Escherichia, the cell surface of Lactobacillus and Bifidobacterium belonging to gram-positive bacteria consists of extracellular polysaccharides, or EPS, and capsular polysaccharides, or CPS, which are highly viscous surface carbohydrate polymers of different sugar compositions and may interact with host immune system. The cell wall also contains a thick peptidoglycan, or PG layer, with the unique components. Several pili structures, which are hair-like appendages and can serve as mucous adhesion molecules, have been reported in both Lactobacillus for the CPA-CBA pili and Bifidobacterium with the sortase-dependent pili and TAD pili. In some Lactobacillus, a glycoprotein structure called S-layer protein, or SLPs, is found on the outside of the cell wall, while Bifidobacterium contains special surface proteins such as serine protease inhibitors, or SERPINs. Additionally, probiotic species may secrete certain antibacterial peptides, bacteriocins, immunomodulatory proteins, and extracellular vesicles. We will describe all of these structures in details in the next few slides. Extracellular polysaccharides, or EPS, and capsular extracellular polysaccharides, capsular EPS, are key components produced by many probiotic bacteria. These polysaccharides play important roles in bacterial adhesion to mucosal surfaces, immune modulation, and protection against environmental stresses. Their involvement in bacterial adhesion and immune evasion is central to their ability to interact with and benefit the host. Extracellular polysaccharides are long-chain sugar polymers secreted by bacteria into their surrounding environment, where they can form biofilms or protective layers around the bacterial cell. Capsular EPS, which are closely associated with the bacterial cell surface, form a dense protective layer around the bacteria. It also contributes to the bacteria's ability to survive in hostile environments, including the acidic conditions of the stomach. Lactobacillus species are well-known producers of different types of EPS, including homopolysaccharides and heteropolysaccharides, each with distinct structural and functional properties. Homopolysaccharides are composed of a single type of sugar unit. In example, Lactobacillus species can produce various alpha-glucans such as dextran, mutan, and ruteran, which are important for forming biofilms or for adhesion to epithelial cells. These alpha-glucans can also influence immune responses by interacting with immune cells. Beta-glucans are known for their immunomodulatory properties, often enhancing the host immune response by stimulating immune cells such as macrophages and dendritic cells. Beta-fructans are fructose-based polysaccharides such as levain and inulin-like molecules. In addition to their prebiotic effects, which are promoting the growth of beneficial gut microbes, beta-fructans also contribute to bacterial adhesion and can help modulate local immune responses in the gut. The majority of Lactobacillus and Bifidobacterium species produce heteropolysaccharides, which are composed of a mixture of different sugar units, such as glucose, galactose, rhamnose, and others. These complex polysaccharides are more structurally diverse and can interact with the host immune system in a variety of ways. Extracellular polysaccharides and capsular polysaccharides produced by probiotic bacteria can play a critical role in modulating the host immune responses through their interactions with toll-like receptors or TLRs. As we discussed earlier, these receptors are pattern recognition receptors or PRRs found on immune cells like macrophages, dendritic cells, and epithelial cells, and they help the body detect microbial components to initiate appropriate immune responses. EPS and CPS can bind directly to TLRs, triggering intracellular signaling pathways that modulate immune cell activity. These interactions often involve changes in cytokine production, the activation of immune cells, and the regulation of inflammatory responses. The nature of the interaction between the polysaccharide and TLRs depend on the specific structure of the EPS or CPS and the TLR subtype involved. For example, some EPS can activate TLR2 or TLR4, leading to different downstream immune effects. EPS and CPS can also indirectly influence immune responses by altering the microbiota composition or enhancing the integrity of the intestinal barrier. This, in turn, can modulate the overall immune environment of the host, potentially influencing the activity of TLRs on mucosal surfaces. Certain EPS and CPS can suppress the expression of these pro-inflammatory cytokines by inhibiting TLR4 signaling. This is beneficial in preventing excessive inflammation. By blocking TLR4 signaling, these probiotics help maintain immune homeostasis and reduce the risk of chronic inflammatory status. This inhibition is likely due to the ability of certain polysaccharides to either bind to TLR4 and prevent its activation, or modulate the downstream signaling pathways that would otherwise lead to the production of inflammatory cytokines. While some EPS and CPAs inhibit TLR4, others can activate TLR2, another important receptor in the immune system. TLR2 is involved in recognizing a variety of microbial components, including peptidoglycans, lipoproteins, and certain polysaccharides. Upon activation of TLR2 by EPS or CPS, immune cells such as macrophages are activated. The activation of macrophages through TLR2 signaling enhances their ability to produce pro-inflammatory cytokines, including IL-6, TNF-alpha, and IL-12. While this may seem counterintuitive in a probiotic context, these cytokines play a crucial role in defending against infections and maintaining the body's immune balance. This figure presents an overview of the effects of EPS on the immune system. In A, EPS can interact by different PRRs like TLRs, Dectin-1, and DC-SIGN. In B, EPS can also influence immune responses by interacting with dendritic cells or DCs and thereby influence subsequent immune responses. S-layer proteins are a unique class of surface proteins found in several lactobacillus species, among other bacteria. These proteins are critical for the interaction between probiotics and the host, playing a significant role in immune modulation, bacterial adhesion, and pathogen exclusion. Here is a detailed explanation of the various aspects of S-layer proteins in probiotics. S-layer proteins are non-covalently attached to the outer side of the membrane of bacteria, meaning that they are not chemically bonded, but instead interact through electrostatic or hydrophobic forces. This allows them to be easily assembled or disassembled depending on environmental conditions. These proteins auto-assemble into a regular crystalline lattice structure that forms a protective layer on the surface of the bacterium. This unique structure provides the bacterium its unique appearance, often described as a tile-like surface visible under electron microscopy as can be observed in this figure. The S-layer serves multiple functions for the bacteria, one of the most important being protection. This layer acts as a physical barrier that shields the bacterium from harmful agents in the environment, including digestive enzymes, bile salts, and antimicrobial peptides produced by the host immune system. The S-layer also helps maintain cell shape, resist osmotic stress, and protect against host immune responses, contributing to the persistence of the probiotic bacteria in the gastrointestinal tract. Many S-layer proteins in probiotic bacteria, including those from lactobacillus species, are adhesins, which are molecules that facilitate the attachment of the bacterium to the surface of the host cells, particularly the intestinal mucosa. S-layer proteins involving adhesions are often designed to recognize and bind to specific receptors on the host epithelial cells or other structures such as glycan, extracellular matrix, or ECM, proteins, or mucins. Another role of the S-layer proteins is in pathogen exclusion. By binding to host cells and tissues, S-layer proteins can block the attachment sites that would otherwise be available to pathogenic bacteria. This process, known as competitive exclusion, helps prevent pathogens from colonizing the gut by occupying binding sites and preventing their adherence. Serpin proteins are a group of serine protease inhibitors that play a vital role in modulating immune responses. In Bifidobacterium species, serpins are crucial for enhancing the survival and persistence of the bacteria in the host gastrointestinal tract. Serpins work by inhibiting the activity of various proteases, including neutrophil elastase and pancreatin, enzymes that are involved in inflammation and tissue breakdown. By inhibiting neutrophil elastase, an enzyme released by immune cells during inflammation, serpins help reduce tissue damage and prevent excessive inflammatory responses that could harm the host tissues. Additionally, by inhibiting pancreatin, a digestive enzyme, serpins contribute to the stability of the gut microbiota, preventing proteolytic degradation of beneficial bacteria. The anti-inflammatory activity of serpin proteins not only aids in the survival of bifidobacterium in the host environment, but also helps modulate the host immune system by reducing excessive inflammation. Pili are thin proteinaceous structures that extend from the peptidoglycan layer of the bacterial cell wall to the extracellular space. They are typically composed of polypeptide subunits which assemble into a long filamentous projections that can vary in length and structure depending on the species and environmental conditions. In Lactobacillus species, pili serves as critical mediators of bacterial adhesion, aggregation, and interactions with host cells, as well as contributing to the overall microbial community dynamics in the gastrointestinal tract. In Lactobacillus pili are primarily composed of pilin proteins, which are subunits that polymerize to form the pili structure. These pilin units are held together by non-covalent interactions and can form a helical or rod-shaped structure that extends out from the bacterial surface. Pili can play a key role in microbe-to-microbe interactions, particularly in the process of aggregation, where bacteria cluster together on surfaces. In the gut, this aggregation can help Lactobacillus species form stable colonies and biofilms, which are essential for persistent colonization and protection from environmental stressors. A well-studied example of pili in Lactobacillus species is the SBA-CBA pili, which are composed of a set of proteins involving adhesion to host tissues. These pili have been shown to interact with host immune cells, including dendritic cells and monocytes through specific receptors. This interaction can activate immune signaling pathways and modulate the host immune response, potentially promoting immune tolerance to commensal bacteria and enhancing immune homeostasis in the gut. In Bifidobacterium species, the tight adherents or TAD and sortase-related pili attach to glycoproteins or glycolipids on the surface of intestinal cells and promotes intestinal epithelial proliferation. These interactions contribute to the maintenance of gut integrity and maturation of the intestinal layer in neonates. Peptidoglycan, abbreviated PG, is a complex reticular structure formed by polymerizing acetylglucosamine and acetylmuramic acid and short peptides consisting of 4 amino acids. In the image shown here, you can observe an example of the polymerizing subunit. In B, it is shown the enzymatic activity of several bacterial enzymes that are involved in the synthesis and degradation of PG. Note that some of the amino acids present in the short peptide can change between microorganisms. PG plays a critical role in providing structural stability to lactobacillus and bifidobacterial bacterial cells, protecting them from external stresses such as pH fluctuations and osmotic pressure. Additionally, PGs exhibit various biological properties, including immunomodulatory effects, anti-tumor activities, and anti-infectious properties. The composition of the pentapeptides and glycan strands is specific to lactobacillus strains. The strain-specific immunomodulation by PGs in probiotics may be mediated by distinct differences in the degree of amidation, glycosylation, and acetylation of PG polymers. Recent studies have shown that PG fragments, also known as muropeptides, can act as signaling molecules to the host immune system and regulate inflammatory responses. Like other bacterial molecules, the PGs act as microbial associate molecular patterns to elicit their immunostimulatory function through the host pattern recognition receptors, or PRRs. They are involved in recognizing PG fragments to activate the downstream signaling pathways in the host, potentially leading to the recruitment of immune cells to the infection site and triggering immune responses. The image shown here provides a schematic diagram summarizing the toll-like receptors, or TLRs, and nucleotide binding oligomerization domains containing proteins, or NODs, in different locations on the cell membrane, cytoplasm, or endosome. As we reviewed earlier, these are the host cell sensors of microbial components on innate immune system cells. Their main activating ligands are shown. I want to show here that NOD1 and NOD2, which bind these muropeptides, activating pro-inflammatory responses. It has been shown that PG metabolites increase the expression of several cytokines and activate pro-inflammatory pathways, so that PG fragments from probiotics have been utilized as natural adjuvants for vaccination in various preclinical models. Proteins secreted by these beneficial microorganisms play a crucial role in beneficial effects of probiotics on host health. These proteins can either be exported to the bacterial surface or secreted into the extracellular space, where they can interact directly with the host and influence various physiological processes. The effect of these proteins contribute to many of the key probiotic traits, such as adhesion to intestinal surfaces, immune modulation, and molecular crosstalk between the microbiota and the host. While much progress has been made in understanding the effects of probiotic-secreted proteins, the precise mechanisms underlying their actions are not fully understood. The interactions between these proteins and host cells are complex and can vary depending on factors such as the type of protein, the probiotic strain, and the specific host environment. Many secreted proteins have pleiotropic effects, and their mechanism of action may involve direct interactions with host tissues, as well as indirect modulation of host physiology through the modulation of other microbiota members. The effects of probiotic-secreted proteins are strain-dependent rather than species-specific. This means that different strains of the same species can produce varying types and quantities of secreted proteins, which can lead to different biological effects on the host. For example, two different strains of Lactobacillus or Bifidobacterium may secrete different proteins that have unique abilities to modulate immune responses, promote epithelial cell adhesion, or enhance gut barrier function. Therefore, the selection of a particular probiotic strain depends on the desired therapeutic effect, and strain-specific differences in secreted proteins should be taken into account when choosing probiotics for specific health benefits. Let's look at some specific examples. Bacteriocins are antimicrobial peptides or proteins secreted by probiotics, including many Lactobacillus and Bifidobacterium species, that have different effects on the host immune response in addition to their antimicrobial action.
Quick question.
Yes.
First of all,
my goodness.
fantastic.
8 a.m. is maybe your sweet spot.
Thank you.
You said that have different effects. Do you want to say direct effects? So including,
Oh, yes. Sorry.
no worries. So to get a nice
Okay.
into that sentence, let's just start at the top and say-
Okay, let me drink a bit.
Thank
Yeah,
you for
so
stopping
take-
because I was trying to hold back up.
But yeah, no, excellent run. That was amazing.
Yes.
Truly spectacular.
Okay, thank you. Direct effects, okay.
Yep. And I'll count us in. Let me know when you're ready.
Okay, go for it.
All right, you can start after 5, 4, 3.
Let's look at some specific examples. Bacteriocins are antimicrobial peptides or proteins secreted by probiotics, including many lactobacillus and bifidobacterium species, that have direct effects on the host immune response in addition to their antimicrobial action. These bacteriocins have been shown to influence the immune system through various mechanisms. Bacteriocins can activate MAPK. No, I want to spell that. Okay.
No worries.
You broke the seal. That's it.
Bacteriocins can activate mitogen-activated protein kinases, or MAPK, signaling pathways, which are crucial for regulating various cellular processes, including immune responses, inflammation, and cell survival. This activation helps the host immune cells respond to signals from probiotics, enhancing the body's ability to mount a controlled immune response. Furthermore, toll-like receptors, which are important for detecting pathogen-associated molecular patterns, are often activated by bacteriocins. This activation can trigger downstream immune responses, influencing both innate immunity and adaptive immunity. Bacteriocins have been reported to stimulate the production of IL-10, an anti-inflammatory cytokine that helps regulate immune responses. Bacteriocins also have the ability to reduce the levels of pro-inflammatory cytokines, such as TNF-alpha, IL-1 beta, and IL-6. These cytokines are involved in the initiation and amplification of inflammatory processes. Other secreted proteins contributing to immune modulation, intestinal health, and epithelial function are, for example, enolase from Bifidobacterium. Enolase is a glycolytic enzyme secreted by Bifidobacterium species that contributes to immunomodulation. Enolase is involved in immune system activation, including the stimulation of macrophages and dendritic cells. It also has been shown to enhance the production of anti-inflammatory cytokines such as IL-10, promoting a balanced immune response that helps maintain gut homeostasis. BOB-A from Bifidobacterium bifidum is secreted and plays a key role in enhancing epithelial cell adhesion. By binding to host cell receptors, BOB-A promotes the attachment of Bifidobacterium to intestinal epithelial cells, or IECs, which is critical for colonization and establishment of beneficial microbial communities. Additionally, BOB-A stimulates the secretion of pro-inflammatory cytokines such as IL-6 and IL-8, which are involved in the regulation of immune responses and inflammation at the epithelial surface. This cytokine production can help recruit immune cells to the site of infection or injury, contributing to local immune defense. Diglycerol kinase, or DGK, is produced by Limoxylolactobacillus reuteri. DGK from L. reuteri is a secreted enzyme that has been shown to reduce pro-inflammatory responses. By modulating the host immune system, DGK helps balance the inflammatory process and promotes gut health. Specifically, DGK can help reduce the activation of pro-inflammatory pathways, protecting the intestinal lining from damage and promoting a more regulated immune response. P40 and P75 are well-characterized secreted proteins from lactic acid-bacillus casei that supports intestinal epithelial cell homeostasis. These proteins contribute to the proliferation and repair of the epithelial barrier, ensuring that the gut lining remains intact and functional. By promoting intestinal epithelial cell proliferation, Pre40 and P75 helps maintain gut integrity and prevents leakage or damage to the intestinal lining. This is particularly important in preventing intestinal permeability and maintaining the gut barrier function, which is vital for immune defense and digestion. Unknown proteins from Lactobacillus acidophilus have been also observed. These proteins produce that, no, wait. I don't know why I put the titles twice, so that's why. Lactobacillus acidophilus produces a range of secreted proteins whose exact mechanisms are not yet fully understood, but some of these proteins have been shown to induce IL-10 secretion in the host. The induction of IL-10 and anti-inflammatory cytokines helps reduce excessive inflammation and maintain immune tolerance, which is beneficial in controlling chronic inflammation and ensuring the proper functioning of the gut's immune system. Probiotics produce a variety of metabolites through fermentation processes, enzymatic activity, and biosynthesis. These metabolites contribute to maintaining a balanced microbiota and modulating host physiology. I will describe the role of some well-studied metabolites produced by probiotic species. The primary short-chain fatty acids produced by probiotics are acetate, propionate, and butyrate. These SCFAs are generated from the fermentation of undigested dietary fibers and carbohydrates by gut bacteria, including probiotics like Lactobacillus and Bifidobacterium species. These molecules have a variety of functions on the host. They are involved in gut health, particularly butyrate serves as an energy source for intestinal epithelial cells, promoting their health and integrity. Short-chain fatty acids can reduce inflammation by inhibiting pro-inflammatory cytokines production and promoting the generation of anti-inflammatory cytokines such as IL-10. Short-chain fatty acids help strengthen the intestinal barrier by increasing the production of tight junction proteins and enhancing the gut epithelial integrity. Short-chain fatty acids influence immune cell differentiation and function, including promoting Treg cell development, which is essential for immune tolerance. As we described earlier, Lactobacillus species in particular are well known for producing lactic acid or lactate as the major end product of fermentation. Lactate lowers the pH of the local environment, creating an inhospitable environment for many pathogenic bacteria while supporting the growth of beneficial bacteria. Lactate has been shown to play a role in intestinal epithelial cell protection by maintaining an acidic pH in the colon and enhancing the mucosal barrier. The proteolysis of dietary proteins results in bioactive peptides which have antimicrobial, immunomodulatory, and antioxidant properties. For example, peptides produced by Lactobacillus species may have inhibitory effects on pathogenic bacteria and antiviral activity. Some probiotic species contribute to the synthesis of several essential vitamins, including B12, folate, and riboflavin, which are important for energy metabolism, red blood cell formation, and overall immune function. Probiotics also release various nucleic acids into the extracellular space, which can interact with both the host immune system and the surrounding microbiota. These include DNA, extracellular DNA or eDNA, RNA, and bacteriophages, and they play important roles in modulating microbial dynamics and host physiology. The role of DNA from probiotic species was reported early on through observation of stimulation of the host TLR9, resulting in anti-inflammatory response. Examples of this are Lactobacillus johnsoni as well as the BSL-3 probiotic mix. Extracellular DNA can be secreted by some probiotic species, including Lactobacillus. eDNA contributes to the formation and stability of biofilms. eDNA can also be recognized by host immune cells, triggering innate immune responses. Evidence is available that indicates that probiotics can secrete microRNAs that may modulate host gene expression. These small non-coding RNAs could potentially regulate immune system function and intestinal epithelial cell turnover. For example, certain microRNAs from Lactobacillus could influence intestinal barrier function by modulating the expression of tight junction proteins. Extracellular vesicles are nano-sized membrane-enclosed vesicles released by bacteria. Note that EVs are also released by eukaryotic cells. In bacteria, EVs are composed of nucleic acids, metabolites, proteins, and lipids. They can also contain lipoteichoic acid or lipopolysaccharides, depending on whether they are released from gram-positive or gram-negative bacteria. This figure is a representation of an extracellular vesicle. While the observation and study in probiotic organisms is rather recent, we already have strong evidence that probiotic-derived extracellular vesicles play a critical role in modulating host immunity. They can promote either anti-inflammatory or pro-inflammatory responses, depending on the composition of the vesicles and the host environment. The interaction of EVs in the host can trigger diverse immune responses, including inflammation through various immune cells, such as macrophages and T cells, thereby aiding in pathogen clearance and modulation of host immunity. For instance, EVs from Lacticacibacillus rhamnosus GG and L-reuteri DSM 17938 have been shown to reduce interferon gamma and IL-17A responses in T cells and natural killer cells, which is dependent on monocytes. Treatment with EVs from Lactobacillus plantarum Q7 has demonstrated alleviation of symptoms in DSS-induced colitis by downregulating inflammatory cytokines, including IL-1 beta, IL-2, IL-6, and TNF-alpha in the gut, and preventing gut dysbiosis in mice. Another example from my lab is the recent finding that Lactobacillus johnsoni and 6.2 EVs stimulate innate immune responses to promote clearance of murine norovirus infections in mice. These are just some examples, but new ones are reported on a daily basis. To summarize, this schematic illustration shows the role of Bifidobacterium or Lactobacillus known effector molecules that play a role in immune regulation. These interactions can have pro-inflammatory or anti-inflammatory effects, contributing to the regulation of gut health, immune tolerance, and immune defense. Some key features in the illustration are effector molecules. Bifidobacterium and Lactobacillus species secrete a variety of effector molecules, such as extracellular polysaccharides, surface proteins, bacteriocins, and exosomal contents. These molecules have been shown to engage with immune cells and modulate immune responses. Immune cell interactions. Macrophages, shown as MO, dendritic cells as DCs, and T cells are shown as interacting with these probiotic effector molecules. Bifidobacterium and Lactobacillus effector molecules may bind to pattern recognition receptors, such as toll-like receptors on immune cells, leading to cytokine production and the activation of various immune signaling pathways. Anti-inflammatory effects. Certain molecules, such as those produced by Lactobacillus, help promote the differentiation of regulatory T cells, shown as Tregs, and IL-10 production, which are crucial for immune tolerance. Pro-inflammatory effects. On the other hand, some probiotic effector molecules may also induce pro-inflammatory cytokines, like TNF-alpha, IL-6, and IL-1 beta, triggering a controlled immune response that aids in pathogen clearance. In this lecture, we define the bacterial components that may have an impact on both the innate and adaptive immune system of the host. The key takeaway points of this lecture are: a variety of bacterial cell components can mediate host immune responses. Microbial components are sensed by specific host pattern recognition receptors. The modulation of host immunity are influenced by the bacterial component and the interacting immune cells. Together, the understanding of the immune modulatory effects of probiotics provides insights into their potential use in promoting gut health and immune regulation. In later modules of this course, we will discuss the effects of probiotics on specific diseases. Done.
Wow, amazing. Truly amazing. All right, take a second to rest after that amazing run, and we'll get you prepped for module 11. This one's only 24 spots.
You could take your shoes off if you want. You know, they are actually
They
comfortable.
look comfy.
It's not that they are not uncomfortable.
I feel good, right?
It's just, it gets you tired speaking. for
No,
so
it's
long.
exhausting. It's exhausting, for sure.
But you made it look easy.
Right.
No, you did. Oh,
You really did.
Well,
That
it's
was-
much easier when I can read and I don't have to think about it. It takes me a lot longer. And I think students get annoyed when you're thinking, when you're talking.
Yeah.
Because nobody has patience.
no, they don't. Don't like their shirt. It's just 2 today, isn't it?
I think the next one is short.
A little bit shorter, as I recall.
It goes from 40 slides to 24.
I think this one has a lot because it's just the animations.
Mm-hmm.
It's not. Yeah, I just finished one yesterday, but the one I have to send today is short. I think Candy wants to get done quicker next week. But don't tell her even she's listening.
Oh, she's listening.
No, I think somebody else was listening to it.
Oh, is she-
Yeah, Rachel.
Oh, Rachel
Rachel,
Patel?
yeah.
Oh,
Yeah, probably Candy is coming a bit later.
oh, that's right, 9.
Okay,
Right.
let's get this done.
Yep.
I have to go make more lectures for Tammy.
All right. I will. Oh, stop the recording.
So I have a question for you on this one. I saw this morning that I think there is one thing that is not correct on one slide, but I'm not 100% sure. Can you switch the slide afterwards?
We can
What,
change
when,
slides.
when you say you're not sure if it's incorrect, is it
I need to correct.
it's content that might not be
Yeah,
correct?
it's something that I think I copied twice the same box text. And I think it may not need to be there.
Okay. Do you happen to know what slide it is?
The
Or I guess that's.
That box that says superoxide dismutase, the second one,
Mhm.
that one.
So
But
if,
that's what I mean. It's just, I think that one is my mistake.
do you want to remove it, or you're saying you need to check it and then let us?
I wanna check it first.
Okay. Yeah, so you just, if you want to check that and then just let us know,
Actually-
we can update it.
I think it should be checked because it's not even.
Oh,
So
I see.
you see, I
Yeah.
think this is. I think I feel comfortable.
Should it
Taking
just,
it
should it
away.
be blank or should it
Should be
read
blank.
something else?
No, it should be blank.
You can do that, and then if you find out that something needs to go in there, we can get it in there after.
Like
But
that.
you will need to take it out from, is it on several or it's just that one?
It's just that one.
No, I feel better if you take it out.
Yeah, so.
Because I'm pretty sure that is, I may just made a mistake.
Yeah, and if you figure out something else that needs to go in there, we can.
Yeah. Because
I don't know. You see, we look at this slide so many times, and you always find
No,
Something.
no, it really is. The number of people who get eyes on things and then, but I even find like, when I'm doing reviews, because I'm often the one who like looks at them the day before, I'm usually I start getting in the zone looking for grammar.
Mm.
And whether images, it's just like people are looking for different things, but even when you're looking for everything, your eye goes right over something
Yeah.
that's obvious.
And even yesterday, that image was there forever. And then yesterday I was like,
Wait a minute.
why did we put this image here? Anyway, Tammy was able to change it. So, okay,
Are you all nice and lubricated with your water?
I am. Is my hair okay to your liking?
I think it is to my liking. We'll know as soon as he puts you on camera, but from here.
I tell you, you're my stylist.
I'm sorry, we're both in trouble then.
You see?
Just a
little more balanced.
So pretty. You're so lucky.
Hmm.
You're so lucky to have such an even curl. Oh, goodness gracious.
All right. Let me know when you're ready. We can give it a whirl.
I am ready.
Awesome. All right. Then you can start after 5, 4, 3.
Hi, everybody. In this module, I will summarize the advantages and limitations of using bacterial and yeast strains. Strains. Strains.
No worries.
That's an easy word.
Once you learn all these hard words, the easy words just get.
They're superfluous. They don't matter anymore.
Okay,
All right.
let's go again.
With that nice big smile, you can start after 5, 4, 3.
Hi, everybody. In this module, I will summarize the advantages and limitations of using bacterial and yeast strains utilized as probiotics for the delivery of health-promoting molecules. I will also briefly discuss the new therapeutic functions in these recombinant microorganisms. It is expected that at the end of this module, you will be able to list the characteristics of Lactococcus, Lactobacillus, and Bifidobacterium that make them suitable as in vivo delivery vectors. Describe the limitations in using Lactobacillus and Bifidobacterium for heterologous gene expression. List the benefits of using Escherichia coli Neisli 1917 and Saccharomyces boulardii as engineered biotherapeutics, and to provide examples of molecules delivered by recombinant probiotics and their applications on disease treatment. We have described until now several
You got through the on-screens. That was great.
We have described until now several of the beneficial effects of probiotic strains on human health. To improve the therapeutic outcomes, these beneficial microorganisms can be utilized as a chassis for the production of health-associated molecules. Taking advantage of synthetic biology tools, these microorganisms can be engineered and the therapeutic efficacy optimized. As a result, engineered probiotics can be used as live diagnostic tools to sense and secrete therapeutic proteins in response to environmental triggers. This image summarizes several synthetic biology approaches in engineering probiotics. The genetic modification of these organisms offer the possibility of therapeutics for the treatment of metabolic and immune diseases. They can be utilized in biosensing and diagnostics. They may be modified for prebiotic usage, improving its colonization and survival in the gut, and they might be used in vaccine delivery. Lactic acid bacteria and bifidobacterium are increasingly being explored as live mucosal vectors for the delivery of therapeutic molecules, particularly for applications in gut health and immune modulation. These probiotics have been identified as promising candidates for the in situ production and delivery of therapeutic agents such as antibodies, enzymes, cytokines, and vaccines directly within the gut environment. Several key features make LAB and bifidobacterium species effective delivery systems. They have GRAS status, meaning generally regarded as safe. GRAS status indicates that certain microorganisms, including Lactobacillus, Lactococcus, and Bifidobacterium, are considered safe for use in food and dietary supplements. This status is crucial for the potential therapeutic use of these probiotics as live delivery vehicles. Their safety profile is well established through extensive studies, which supports their application in human health interventions. LAB and bifidobacterium species can be engineered to produce therapeutic proteins and bioactive molecules in the gut, where they can actively interact with the host immune system or intestinal microbiota. The idea is that these live bacteria will produce the desired therapeutic molecules locally within the gut as they traverse through the gastrointestinal tract. This approach can reduce the need for systemic delivery, providing a targeted effect at the site of disease. Other factors that enable LAB and bifidobacterium species to function as effective delivery systems is their ability to survive and persist under the harsh condition of the gastrointestinal tract. These organisms must be able to withstand the low pH in the stomach. LAB and bifidobacterium species have evolved mechanisms such as acid tolerance proteins that allow them to survive and remain viable throughout this acidic environment. The presence of bile in the small intestine can also be toxic to many microorganisms. However, LAB and bifidobacterium species are known to have bile salt hydrolase activity, allowing them to tolerate and survive in bile-rich environments. Under these environmental conditions, Lactococcus lactis is known to survive for up to 48 hours in the gastrointestinal tract, while Lactobacillus and Bifidobacterium species can survive between 4 and 7 days, respectively. Lactococcus lactis is one of the best studied lactic acid bacteria, particularly in terms of its physiological properties, genetic manipulation, and use in mucosal delivery system. It is widely recognized model organism for probiotic applications, especially for the production and delivery of therapeutic molecules in the gastrointestinal tract. Some of the key characteristics of Lactococcus lactis are: Lactococcus lactis is extensively studied at the physiological level, providing a solid foundation for its use in biotechnology and health applications. Its genetic, metabolic, and fermentation pathways have been mapped, making it a reliable model for studies on metabolic processes, enzyme production, and biotechnological applications. The genome of Lactococcus lactis has been sequenced and several complete genomes are available, which has facilitated genetic engineering and the development of tailored strains for various therapeutic applications. Genetic tools such as plasmids, expression systems, and vectors have been developed, enabling the transformation of Lactococcus lactis with therapeutic genes and the expression of bioactive molecules like cytokines, enzymes, or vaccines. Lactococcus lactis is known for its high transformation efficiency by electroporation, a process that allows for the easy introduction of foreign genes into the bacterium. This is important for the engineering of Lactococcus lactis to produce specific therapeutic molecules for mucosal delivery. Another advantages of Lactococcus lactis is the simple secretion system, which enables the efficient secretion of proteins or therapeutic molecules into the extracellular environment. This system can be used to deliver therapeutic agents directly to the mucosal surface in the gastrointestinal tract, making it an effective vehicle for the in situ production of bioactive molecules. The NICE system is the most widely used protein expression system for Lactococcus lactis. It is based on the control of strong niacin-inducible promoters like P-NIS-A or P-NIS-Z, and two of the cognate signal transduction NIS-KR two-component system. The integration of the NIS-KR genes downstream of the PEPN gene in the prototype strain MG1363 yielded the Lactococcus lactis NC9000 strain. Since then, Lactococcus lactis, oh, you're going too fast.
Sorry. We can start it since then.
Okay. Since then, Lactococcus lactis NC9000 has been distributed to laboratories worldwide and has become the laboratory workhorse for protein expression and has been used in many physiological studies. Lactobacillus species have also shown considerable promise in the context of mucosal delivery system. These bacteria possess numerous probiotic features that make them excellent candidates for therapeutic applications in the gastrointestinal tract. However, challenges such as genetic manipulation remain significant. Some key advantages of Lactobacillus as a delivery vector are: many Lactobacillus species possess probiotic properties, including immune modulation, gut health maintenance, and inhibition of pathogen growth. These characteristics make them effective for applications such as gut-targeted therapy or immune system regulation. Lactobacillus species can persist in the gastrointestinal tract for longer periods than Lactococcus, making them more suitable for long-term therapeutic applications. Some species can survive for several days in the gut, as we just described. This is linked to the ability of survive harsh gastrointestinal conditions. Several Lactobacillus species are already part of the native gut microbiota in humans, meaning that they are naturally adapted to the intestinal environment. Since Lactobacillus species are part of the normal microbiota, they are less likely to provoke an immune rejection or unwanted immune responses compared to non-native organisms, making them ideal for therapeutic use. These interactions also make Lactobacillus a promising tool for applications in vaccine delivery, where the bacteria can help boost the mucosal immune response to targeted pathogens. While Lactobacillus has many advantages, one significant drawback is the difficulty associated with genetic manipulation and transformation. Unlike Lactococcus lactis, which has relatively straightforward transformation protocols, Lactobacillus species are notoriously difficult to genetically manipulate. This difficulty arises due to the challenges in DNA uptake, transformation efficiency, and the lack of efficient genetic tools for certain Lactobacillus strains. While genetic systems have been developed for some Lactobacillus species, the lack of universal system for all strains limit the ability to engineer these bacteria for targeted therapeutic delivery. This is a significant barrier for the widespread use in genetic-based applications, such as the production of therapeutic proteins or gene delivery. In a later module, we will see a more extensive list of the therapeutic potential of recombinant probiotic strains. However, with this figure, I want to show some examples of molecules that can be engineered with beneficial effects. Recombinant lactic acid bacteria, shown here as REC-LABs, can deliver widely diverse molecules in the intestine, where they exert various health benefits. In A, the oral administration of a recombinant LAB expressing elafin prevents colon inflammation in mouse models of acute or chronic colitis by decreasing elastoic activity and restoring intestinal homeostasis. Furthermore, REC-LAB causes a decrease of intestinal permeability and reduced cytokine and chemokine release from inflamed epithelium. In B, engineering lactococci and lactobacilli secreting the antioxidant enzyme superoxide dismutase, shown as SOD, decrease cecal and colonic inflammation and lead to an anti-inflammatory effect in murine model of colitis. In C, Lactococcus lactis can deliver plasmid into eukaryotic cells, resulting in the epithelial cell producing a protein of interest. In D, treatment of NOD mice, a model of type 1 diabetes, with intravenous injection of anti-CD3 and oral administration of IL-10 and pro-insulin secreting LAB, induces a long-term remission of type 1 diabetes. This combination therapy increases the frequency of local and functional Treg cells and leads to the restoration of antigen-specific tolerance. This last therapy is already in human clinical trials. Bifidobacterium as a delivery vector enjoys the same benefits and limitations as those described for Lactobacillus. Bifidobacterium are abundant in the human gut microbiota, particularly in infants and children, and they contribute to intestinal homeostasis. Their native presence in the gastrointestinal system makes them particularly suitable for mucosal delivery, as they are already well tolerated by the host and can establish interactions with intestinal epithelial cells and immune cells. Despite the promising potential of Bifidobacterium as delivery vectors, there are several challenges that need to be addressed. Bifidobacterium species generally have low transformation efficiency compared to other bacteria like Lactobacillus or Lactococcus. While genetic manipulation systems for Bifidobacterium have improved, they are still more limited when compared to Lactobacillus or Lactococcus. The development of more efficient plasmids, expression systems, and gene editing tools specific to Bifidobacterium is still in progress. In addition, Bifidobacterium species tend to have low stability when it comes to maintaining genetic modifications, particularly when using episomal plasmids. Integration vectors that allow for stable genomic integration are being developed, but the challenge remains for long-term gene expression and protein production. One example that I want to highlight is the use of Bifidobacterium longum as a live vaccine. In this strain, the BP1 protein from Enterovirus 71 was genetically engineered and expressed in Bifidobacterium longum. Escherichia coli in Italy in 1917 is a well-characterized non-pathogenic strain of E. coli with a long history of use as a probiotic. It has shown significant promise as a delivery vector for therapeutic applications, particularly in immunotherapy, gene therapy, and vaccine delivery. This strain possesses several probiotic characteristics such as the ability to restore intestinal microbiota balance, enhance intestinal barrier function, modulate immune responses by inducing the secretion of anti-inflammatory cytokines like IL-10, and suppressing pro-inflammatory cytokines. This strain has proven safety in humans. E. coli Nisli 1917 is classified as non-pathogenic and has been used therapeutically in humans for over a century. The strain has GRAS status approved by the FDA, making it safe and reliable candidate for oral delivery of therapeutic agents. Another interesting feature is that unlike other non-native bacteria, E. coli Nisli 1917 can persist in the human gastrointestinal tract, especially in the colon. It naturally adheres to the intestinal mucosa and interacts with the host immune system. The strain's ability to persist in the gut and form stable colonization allows it to sustain therapeutic effects over prolonged periods. E. coli Nisli 1917 offers several advantages as a delivery vector. It has high efficiency of genetic engineering. E. coli Nisli 1917 is highly transformable and it's amendable to genetic manipulation. This strain can be efficiently engineered to express and secrete therapeutic proteins such as cytokines, enzymes, or antigens. A variety of genetic tools such as plasmids, CRISPR-Cas9, and expression system makes it easy to insert genes encoding for therapeutic molecules. E. coli Nisli 1917 has well-established gene expression systems, including both inducible and constitutive promoters for precise control over the production of recombinant proteins. Additionally, the strain possesses the ability to secrete proteins into the intestinal lumen, facilitating local delivery of therapeutic molecules directly to the site of action in the gastrointestinal tract. E. coli Nisli 1917 has been used as a vaccine delivery vehicle, delivering antigens from pathogens directly to the gut-associated lymphoid tissue, or GALT. Some in vitro as well as animal studies have shown that E. coli Nisli 1917 can trigger robust immune responses, making it a promising candidate for oral vaccines targeting pathogens such as Shigella, E. coli, Salmonella, and rotavirus. Another example is their use as an enzyme replacement therapy. One example is SINV1618, which has been engineered to encode for phenylalanine metabolizing enzyme as a treatment for phenylketonuria. This LVP has completed phase 2 clinical trials. There are several challenges of using E. coli Nisli 1917 as a delivery vector. While this strain is considered safe for use in probiotics, its application as a gene delivery system or therapeutic vector may still face regulatory hurdles. The use of genetically modified organisms or GMOs in human therapies is highly regulated, and ensuring the safety and effectiveness of such treatments will require extensive clinical trials and regulatory approvals. Although E. coli Nisli 1917 is non-pathogenic, there remains the possibility of triggering immune responses due to the presence of certain surface molecules, in example, lipopolysaccharides or LPS. The immunogenicity of these components could lead to a mild inflammatory responses in some individuals, especially in the case of systemic exposure. One of the challenges with using E. coli Nisli 1917 as a delivery vector is ensuring the stability of genetic modifications. The strain's plasmid-based system may not always maintain stable gene expression, particularly in the intestinal environment. Integration systems to stabilize genetically engineered strains are being developed, but plasmid loss and instability are ongoing concerns. While E. coli Nisli 1917 is capable of persistent colonization in the gastrointestinal tract, the balance between long-term persistence and immune clearance remains a critical factor. Over time, the host immune system might eliminate the transformed strain, reducing the effectiveness of the delivery vector. This can be especially relevant in clinical applications where sustained gene expression may be required. Saccharomyces boulardii is a non-pathogenic yeast strain. It is gaining attention as a novel biotherapeutic delivery vector due to its unique properties, probiotic characteristics, and potential to be engineered for mucosal delivery of therapeutic molecules. I want to highlight some of the key features and advantages of Saccharomyces boulardii as a delivery vector. S. boulardii is GRAS, generally recognized as safe by the FDA, making it a safe candidate for human consumption. It is resistant to gastric acid and bile salts, allowing it to survive and persist through the harsh condition of the gastrointestinal tract, including the stomach and small intestine. S. boulardii has the ability to adhere to the intestinal epithelial cells and intestinal mucus, promoting its persistence and ensuring that therapeutic molecules can be delivered directly to the gut. Saccharomyces boulardii can be engineered to express and secrete therapeutic molecules such as cytokines, enzymes, antigens, or antibodies. While S. boulardii is a non-conventional yeast for therapeutic delivery, it is genetically amendable to modification. Genetic tools for S. boulardii are derived from its close relation to Saccharomyces cerevisiae, a widely studied yeast. It can be transformed using standard techniques like electroporation and can be engineered to express foreign proteins, making it a versatile vector for therapeutic delivery. Recent advances in CRISPR-Cas9 and other genetic modification systems have improved the efficiency of engineering S. boulardii for therapeutic purposes, including antigen delivery, cytokine production, and enzyme secretion. Some of the therapeutic applications of engineering yeasts are: in oral vaccination, S. boulardii can be engineered to express antigens from pathogens such as Salmonella or Clostridium difficile. In gene therapy, S. boulardii can be used as a gene delivery system for therapeutic applications, such as delivering therapeutic enzymes for metabolic disorders or immune modulating proteins for diseases like IBD and colitis. In the treatment of gut disorders, S. boulardii has been shown to reduce the severity of intestinal diseases such as antibiotic-associated diarrhea, C. difficile infection, and inflammatory bowel disease. When engineered to express therapeutic proteins such as cytokines or digestive enzymes, it could offer therapeutic benefits by restoring gut homeostasis and intestinal integrity. There are some challenges with the utilization of Saccharomyces boulardii as a biotherapeutic delivery vector. While S. boulardii is genetically manipulable, its genetic manipulation tools are not as well developed as those for bacterial systems like Lactococcus lactis or E. coli. The development of more advanced genetic engineering techniques for S. boulardii is an area of ongoing research. Can you back them?
Sorry, what are you?
No, that's okay. I'm going to talk.
Okay.
Some of the general regulatory concerns are linked to the use of genetically modified organisms, including genetically engineered S. boulardii. Like other GMOs, the yeast used as a live biotherapeutic will need to undergo extensive clinical trials to confirm its therapeutic potential. Although S. boulardii is generally regarded as safe, immune responses to its yeast components, such as beta-glucans or mannan, could occur in sensitive individuals, particularly with genetically modified strains that express foreign proteins. Careful monitoring and safety assessments will be needed. In this lecture, we define the use of engineering probiotics for the delivery of health-related molecules. The key takeaway points of this lecture are: lactic acid bacteria and bifidobacterium species are promising candidates due to their GRAS status, ability to survive the harsh gastrointestinal environment, and their potential for in situ production and delivery of therapeutic molecules. Lactococcus is the preferred LAB due to the genetic toolbox available. Lactobacillus and bifidobacterium species have low amenability to genetic manipulation. Escherichia coli Nisli 1917 and Saccharomyces boulardii have GRAS status with the FDA and can be genetically manipulated for the delivery of health-related molecules. The main limitation of engineering probiotic strains is their GMO status, which warrant further regulatory and public acceptance challenges. Done.
Nice.
Okay.
There we go.
Bravo.
Are we done yet?
We are. All right.
Now I have the whole day to work.
Oh, you do, yes.
Are you meeting with her after this, or you're just going?
I'm not sure if she's available.
She's here. She got here.
Yeah, I don't know. I think she said she had a meeting at 10:30. I'm not sure. If not, just.
Oh, do you want to bring her back to my office? Yes. I will do that.
Very good.
Okay.
Good job today.
Okay.
It was
thank you.
lovely.
Thank you for your help.
What help? What help? You did
We
great.
just sat back and watched. You knocked it out.
Okay. I
See,
think we
mornings
have
are
the-
good maybe, but I know, the drive. I know.
Enjoy your Thanksgiving break.
You too. Good luck with your cooking.
Thank you. You
No,
as well.
You're gonna have more fun than we are.
Oh,
Thanks.
you always do.
Bye.
Bye. Have a good one. Whichever way you want.